For the next round, we'll have a great series of talks now looking at applications of these technologies, and some pretty fascinating and interesting ones. We're all super excited to have Axel here to talk about his project. It takes a really somewhat novel approach to this idea of printing and of fabricating, using microfluidics in a really brilliant way. And I'm just going to learn. Thanks a lot. I really appreciate you coming out.
Thanks. All right. Very happy to be here. It's been very exciting. I'm going to talk to you about bioprinting in a little bit different sense, that we use microfabricated devices to define sheet and tubular constructs with cells in them. We just had demonstrations with alginate. We're going to see a little bit more alginate and cells in alginate. But I'll start different in a sense that I start from an intact organ, because when I got my training in Europe in my PhD in engineering, and then I did some work in Boston in microsystems for chemical synthesis, and I went to Toronto, and now in Toronto, where there's an abundance of biology-minded people that are very hard to avoid, and I got really interested in using microsystems for applications in biology. And I'll start with an intact organ, which is a blood vessel. So next slide, please. So we go from organs to cells, and I say not quite back. And so that's the second part is bioprinting. The first one is characterizing some. I think so. The next slide. The first one is in sticking some of these soft intact tissues in a microsystem and characterizing them in a way that is very similar to the in vivo environment. And the one reason why I put this up is because it's so challenging to manipulate these vessels. So these are 200, 300 micron diameter blood vessels taken from an animal, about 1 millimeter in length, and you try to keep them in a way that's similar to what they're experiencing in vivo. And that challenge is the same whether that is a bioprinted tubular construct or whether it's an intact vessel. All right, so we call this artery on a chip, and you see a lot of people have contributed. And this is meant to say artery chip, right? And it's not a commercial, near commercial product, so it's technology. In the next slide, you'll see that allows us to study intact resistance arteries. And so it's a cross section of one of these vessels. And so when you think about bioprinting, we think it's a challenge to put the right cells in the right place. Maybe the endothelial cells are the ones that are meant to be around the lumen of the cell here, and then smooth muscle cells around. But there are many more challenges. And there's also the challenge of putting the right extracellular matrix molecule in the right alignment, not just the right material in the right space, but also the right material in the right organization. And so it can actually hold the blood pressure. None of the bioprinted constructs right now, tubular constructs, can be immediately pressurized after printing, right? It's just not possible. So if you go to the next slide, so it's very challenging physiologically. So one thing we've done is we built a little gadget that is 1 by 3 inch in size out of plastic, and that you can stick one of these vessels in there. You could also stick a bioprinted vessel in there, and you keep the temperature as physiological temperature of 37 degrees. But that's not it. You need to be able to also pressurize it, at physiological pressure levels. And so there's different colors here. In green, that's what goes through the innermost part of the vessel, the lumen. That will be pressurized, and these forks at the end, those are the ones that allow us to keep the vessel in place. Right? We apply a sub-atmospheric pressure. I stopped calling this a vacuum because some biologists thought I'm going to refer to gas environment. So this is all liquid filled. All these channels are liquid filled with culture medium, but the yellow forks are at the subatmospheric pressure. And now what this allows us to do is address the luminal and the abluminal side of these tubular constructs separately. And so we can now stimulate these vessels with drugs, we can measure diameter changes, and we've done that. And we go to the next slide. And now if you're interested in playing with something like this, then there is a commercial version. You can stick this onto a microscope and you can basically use it. Go to the next slide. And now we're a bunch of geeks and engineers in the lab. We'd like to build systems that are fairly automated, and beyond sticking in the blood vessel onto a chip, we like to also automate the characterization. And if you're like-minded, you may like this idea. So you could put a vessel on a chip and then sort of go home or fly somewhere to a workplace, and let the computer do all the work for you in terms of characterizing using immunohistochemistry of different types of markers. Go to the next slide. So this is now how the gadget looks like. This is probably something that, depending on your comfort zone, that makes you either love the technology or make you very unfamiliar with it. On the right side, I show you the chip design. This is the architecture that is needed, fluidic architecture needed to keep the blood vessel at this small spot. This is 1 inch by 3 inch, and here is where we're going to stick it. Okay, go to the next slide. And so now what I show you is a chip-hosted live vessel that's in this field of view that is pressurized. There's a brain vessel from a mouse. And what we're going to do is we're going to subject the vessel on the outside to an increasing level of a drug that makes the vessel constrict. So if you go to the view forward, you see that the diameter changes, right? And you can go further and further, right? So this is the diameter change as a function of the applied the drug concentration. You see there's a very significant diameter change, right? If you're a tissue engineer or a bioprinting expert and you look for a challenge, then one good challenge is you print a vessel that can do the same thing, right? That can cause a diameter change, so let's say 20, 30%. And no one can do that. Anyhow, but so if we go back to the next slide, so this was, I think you cycled back through the sequence. It's a very drastic diameter change, and that is an indication of this vessel being functionally intact. So this is how the machine looks like, right? Or makers, right? This is sort of a piece of engineering, right? A lot of software needed to address it. Go to the next slide. And then we've used it to carry out different types of staining protocols that are proving us that this is a functionally intact blood vessel. Go to the next slide. Yeah, good. I think we can go to the more staining. Okay, so now I wanted to spend most of the time talking about the other direction, right? So going back from cells, not quite, to organs, right? And this is also where some of the challenges come into play, or opportunities, however you want to look at them. So we talk about bioprinting from a little different angle. Go to the next slide. I carry a German passport, so when I think about printing, I think about this fellow here, and he was the one who invented the printing press a long time ago. It's not around anymore, and he was able to do 240 impressions per hour on his press, right? And the way this works is you have a substrate, which is paper, and you put something on it, which is ink, right? And nowadays, there's two versions of this. One is a very high throughput system that's used for making newspaper, for instance, or books. Or there's a reasonably low-cost system that every one of us has on their desk. And this one is sold not by selling a box, but by selling cartridges that we all pay for, and we don't like it, but we still do it, and that we regularly need to refill. Okay, so you go to the next one, and now we do know that. So why I elaborated a little bit in detail is that, of course, for bioprinting, current bioprinting systems, none of the two really applies, right? So we're developing, or we have developed a bioprinting system I'll show you in a minute. And so this is an audience that's well aware of the fact that there's all kinds of different organizations and tissues, and I'm particularly interested in layered tissues. And so I always start with a leaf, and I was extremely happy to listen to some of the previous talks. I'm extremely unsuccessful to recruit a graduate student that's, as excited about leaves. Even though before the slide, we're in Canada, so you think someone should be very excited about printing a maple leaf. So, we're mostly working on applications that relate to skin, some cardiac microtissues, as well as blood vessels. So, what we look for is a way that allows us to, with simple means, very scalable means, to create heterotypic organization and soft materials as well as cells that's similar to these tissues. Go to the next slide. And so there's lots of the technology available that allows us to co-localize cells and particles and fibers, and to go further, and then organize these fibers or modules up in planes, and then assemble planes into multiple planes. But none of this really shows up in a significant number in an assembly that's really organ scale. Instead, surgeons are really excited about decellularized organs, right? And so there's a little bit of message there. And we thought one item that's definitely missing is scalability. So we want to have a simple technology that's scalable. And I'll show you how this now works. By the way, some of the materials, and we talked about alginate that we're using, there's all kinds of molecular astronomy outfits. The Canadian one is Molecule R from Montreal, and we've been using their alginate. And whenever we get close to publication, we kind of want to back up some of the molecular R alginate data with some properly characterized alginate, and realize that some of these data are hard to reproduce, and this alginate actually works quite well. And but there's a long list of extracellular matrix materials of polymers that are extremely well understood and characterized in the biomaterials community to choose from. Okay, if you go to the next slide. So you start the animation. So I'll briefly discuss how our system works. You can further animate. So if you imagine if you participated in the demonstration of alginate cross-linking just a couple of minutes ago. It's nothing else. As you imagine, we have two pots, could be more, and they contain two types of biopolymer solutions, or two types of identical biopolymer solutions with different cells. One is blue, one is red. And now you'd like to form a sheet out of them, and you'd like to also make sure that the sheet is heterotypic. So you want to have this ability to control at which location you put cells or you don't put cells, and perhaps also what cell type. Okay, so if you further animate, and you'll see that at the beginning, so we have a microfabricated device that I don't show you here, right? I just schematically show you this box, and I'll show you in a minute how it actually works. So you have these two solutions flowing through a device and then exiting something that you can imagine as a sort of a lateral nozzle. So it's laterally distributed access device. A cross-linking starts if you continue animating, and then we can control the topology and its organization. Since we're living in Toronto and we didn't have another bright idea, we just started writing, suggesting we could write Toronto. But you could also think about creating materials that have gradients or patterns with different stiffnesses or diffusivities or different embedded factors. And so this is a continuous process, and what enters here is a fluid, what leaves here is the solids, the sheet that you can keep collecting. Right? So if you continue to animate, then we literally collect this. And the first iteration was a Lego Mindstorm type of technology. That's just driving the drum here. Now the actual chip that we're using is microfabricated using common fabrication technologies, nothing really too particular about it. And this is a collection drum. And so we can control the thickness of these sheets. We can make sure we don't want to make too thick sheets, right? So good thicknesses are 200 to 250 microns to make sure cells can't enter the core are well nutrient supplied. And this is sort of the more elaborate version of the sheet that allows us to create heterogeneity and introduce cells at different locations. I'm not going to explain in detail, but what we do have are small valves that are filled with biopolymer solutions containing cells, and with pressure control we can actuate them or not actuate them. Every time the pressure is up, we inject a small amount of cells into the sheet before cross-linking. So if you go to the next slide, this is how this system works. So there is biopolymer entering here. There's a microfabricated device. This initially liquid sheet exiting here, being cross-linked while it exits, and then collected here in a drum. Moving place a little bit slowly, but this is approximately 1 inch, and the collection or the extrusion velocity is somewhere between 1 to several centimeters per second, so relatively fast. And we've made sheets that are much wider, right? So you can make very large aspect ratio sheets if you want to. The rate limiting step now is not really throughput, it's cell supply, right? So, we can, every time there's lots of cell biologists in the room and I start thinking loud about how many cells per second or per hour or per day, then there are stem cell people, they kind of walk towards the exit of the room because it's a lot of cells, billions of cells. Okay, so this is how the heterogeneity is being generated. So we have these little wells that are pressure modulated, and we collect at the exit. So initially, if you keep animating, we wrote Toronto, as I suggested, right? And I want to get a patent for this. The university told me this is completely useless research because, I wrote Toronto and said, well, I can write it in binary, right? Isn't that good? and I can eat it afterwards. Isn't that even better, right? It's not useful. So that I was thinking Gutenberg, but the student who's working on this has suggested this would not be worthwhile pursuing. So the compromise solution was we wrote the first article, first chapter of the UN Charter in binary code. If you go to the next slide, this is how it looks like. It says to maintain international peace and security, and to that end, and so on. So it's all in binary form, and it takes 88 centimeters, so this length of alginate, and a couple of minutes to write. Had we gone for the Latin option as the Bible, right? This just this would be 1 month to do 2.5 kilometers text and 2.3 liters of alginate. Still manageable, right? So from a cost perspective, it's very manageable. But there was a lot of opposition from the students for a good reason. Okay, so we go to next. And we put this in the supplement of the paper to just be fair. So since I was told that this is not really a fruitful line of research, and we have this abundance of excellent tissue engineers just around the corner, I started to collaborate with a colleague of mine, Melissa Radicic, who's an expert in tissue engineering for heart. And so we put some primary cells in there, cardiomyocytes and fibroblasts that are co-localized. We keep animating. Then we show that these cells are indeed populating the entire sheet, not just the top surface. If you go to the next slide, you'll see that they're healthy. We can create different types of patterns, right? And if you keep and they nicely attached to the matrix, right? So in this case, it was not just alginate. The alginate contained a little bit of collagen, and these are viability data, excellent viability data for up to 15 days. And if you continue, we could also stack these sheets, right? So you could make multiple layers. And if you go further here, We could make bilayer sheets. You could wrap them around. They look like tissue duct tape. Now they, at this point, don't contain any cells. We could introduce some primitive form of vascularization by, instead of introducing the second material here, just introducing void spaces. If you keep animating, we have also wrapped these sheets around themselves to sort of make tubular assemblies. They look like they want to become blood vessels at some point in the future, but they really aren't at this point. Right? So if you keep, so what we're using this technology for right now is for the printing bilayer sheets with skin cells, right, where we have, if you go to the next slide, where we have sort of select regions that are self-populated and others that aren't, that we can culture in vitro and then place on an animal. So these are human cells, but you can place them on a nude mouse. You can skip this slide, and there's some data that show that, as expected, the cells attach nicely within these spotted areas to the matrix. If you go to the next slide, we have, and this is what I wanted to end with. We've adapted this technology that's essentially a nozzle that allows you to extrude a sheet into a slight modification that is a nozzle that has, it extrudes into a borehole, right? That's an engineer talking, right? So in vertically down, these are sort of footprints of the channel pattern that makes it work, which you may not need to really understand in all detail. But if you go to the next slide, then there's a cross-sectional view up here. It kind of shows how it works with a microfluidic device. Make sure that radially biopolymer solution approaches a central hole, and then we extrude tubes down in the vertical direction. We can control these tube diameters by controlling flow rates very reproducibly. And if you go to the next slide, we can make all kinds of beautiful tubes, strange tubes, right? We can make crimped tubes. Physiologically, there's a reason for being interested in crimped tubes. We can make tubes that have stripes in them. We can have single, multi-layer tubes, very reproducibly. We make half-half tubes, Janus tubes, right? Whatever you like. So if you go to the next one, we can make collagen tubes, and this is really something I'm extremely happy with. If you, and you can print the tube, you don't have to wait, right? You can print the tubes there immediately, right? And now you can stick a bubble into this tube. So at the beginning, I wanted to get some proof it's actually an intact tube, right? So show me the tube is intact. You can refuse it, okay, but stick a bubble in there and then manipulate with pressures left and right. This movie may not play, and show that it actually works. So there is the, if you go to the next slide, or maybe it does. Yeah. So now there's gentle manipulation of this bubble in this direction, that direction. You get a feel for it. This is a, for anyone who has a feel for the elastic moduli, a very soft tube, tens of kilopascals of elasticity, and you see that the wall adapts, right, as soon as you manipulate the pressure. And so it's a very enjoyable movie for me to watch. And if you think about that further, the diameter change here, if you know what the surface tension of the bubble is, diameter change, that's you measure elasticity. If you go to the next slide, so the students working with this, there's a vacuum cleaner inventor in Britain that's the name Dyson. He has a design award that he awards every year. Last year, I don't know, the 4th, 5th, or 6th generation of our printers that get smaller every time you build a new one got one of these awards. And so we think that's basically, this is a well plate, right? Twice the size of a well plate could be your BioPrinter, right? And you can try to build an even smaller one. And the throughput is very large. Okay, so before I end, there is, of course, if go to the next slide. This is sort of in Toronto, right? Toronto, Canada, nice place. If you don't know what to do with your time, in June 15 to 18, right? We have the BioPrinting Summer Institute where there's a bunch of lectures, and not unlike here, right? Hopefully as well organized as here, right? And you're welcome to come, right? If you go to the next slide. So this is CN Tower in Toronto. It's 550 meters tall. And starting this year, you can sort of do what these guys do on the outside. Right? if you have some bioprinted material that you believe in very strongly, you can make yourself supported there. Right? So the next slide. So these are some of the guys who were involved in this and helped not just coordinating it, but contribute guest lectures. And I wanted to end on this note. Thank you very much. If you have any questions, happy to.
What was the wall thickness of that tubular bubble?
The tubular bubble, it's somewhere between 100 and 150 microns. Wall. Yeah. Diameter relatively large. So this is these are 1 millimeter glass capillaries for cannulation. So you see it's like 2 millimeters. But they could be. There's no reason to believe they couldn't be made smaller. There's some fabrication limitation in the devices that we're currently using that lets us favor around 1 millimeter, 1.5. But it could be smaller. Could be bigger. Right?
So what kind of pressure do you think you could support in your best tube that you could?
So these, well, in terms of printed tubes, and now this is my personal opinion, I think there's two, in the near term, right? So there's two types of ranges of architectures. One is like this, which is very soft, right? Because the soft is good because, it's what cells like to see. The other is, and challenge would be to actually see something like a constriction, maybe in a tube like that had some cells that were circumferentially lined. That would not be a tube that can take a lot of pressure, right? A tube that can take a lot of pressure for a large blood vessel would need to have alignment of this extracellular matrix in a circumferential direction, and this is something I can't yet talk about. But they can also be printed. And I'll tell you more next time I come. Physiologically, the way this is done, of course, in a real blood vessel, and that goes back to one of the earlier slides, is that you have a real vessel can do both, right? It has locally, it's very soft, and then if you apply a lot of pressure, it can still take it. The way it does it is with the crimped architecture, right? So it has alignment, but there's a crimped molecular architecture around the circumference. And now replicating this by printing goes way beyond, just having a layer of the right extracellular matrix molecule, maybe a thermally cross-linked collagen or something like that, right? So for the time being, I think there's two different interests, right? One is keep the cells happy and get maybe some functional response in terms of constriction, which is soft material, and then at some point the crimped, highly aligned one that could be used as a graft. Anyway, so that's other questions?
I think when you're bringing cells out, I mean, is there that much shear that you have to worry about keeping them alive from the actual process, or it's really just keeping them in the right, in a gel that they're happy in?
Yeah. So the amount of shear is very low in our process, definitely lower than any inkjet system, right? The constraints are also with respect to extracellular matrix choice. The constraints have to do with cross-linking speed, right? Alginate now is an example of something that cross-links rapidly. If you thought about collagen, right, then it's more challenging. But I've shown you these collagen tubes, right? So in that case, we haven't made thermally cross-linked collagen sheets. But for tubes, you just need a larger confinement length, and you can make them. It's also a slower process because it just takes, gelation takes time.
How many layers will you need to print to print the structure to replace blood vessels? And how many layers are involved in a blood vessel. This, I think, you've done 2 layers.
Yeah, but the question, the question is, what is the layer in it? So you could, how many layers does the blood vessel have, right? You could say, well, it's 2 layers when you talk about the different cell types, 2 or 3 layers, right? Say you want to have endothelial cells in the core, and then you want to have some smooth muscle cells wrapped around, right? But then you could look at it in greater detail, right? You could say, well, how is the extracellular matrix organized, right? And then from that aspect, you could say, well, I want to have more layers because I want to replicate the way the material, the protein structures actually.
But how much of that self-organizes because of the phenomena that we've already talked about earlier in the day?
Which is the cells to itself?
Yeah.
Well, yeah, so that may be true, but you also have to see if you printed let's say you print a tubular construct and the cells love it, right? And you keep it in a dish and you culture it for a couple of days, what would happen, right? It would probably be some blob, right? Which doesn't mean you printed the wrong thing. It doesn't mean the cells are not doing what they're supposed to do, but it means that in the body, the tubular or the blood vessel, there's a counteracting force, right? So it's under pressure, right? So you need to replicate that, and initially in some kind of a perfusion bioreactor, right? Initially at probably low transmolar pressures, then maybe over time you can up that. The reality is only that this takes a long time, right? And so for many applications, you may not want to spend that time, several weeks to months, right? So if you're interested to print, and this is kind of often suggested, right, but not really yet possible, right, that you could print and soon after be able to subject the tube to physiological pressures, for instance, right? So that is one of the challenges. But I guess it's good to have some challenges.
The flat pieces, there's the roller pulling that ribbon through. With the tube, is there a pulling force, or is it just pushing out the?
Yeah, it's just hydrodynamic flow. There is a confinement. There's the tubular confinement. Everything is guided through, right? And we've shown that this is actually very, very helpful to have. But we don't collect as in the other case. And if you did, then the question is, how would you do that without collapsing the vessel, right?
Apologize if I missed this. So the printed tubes compared to the blood vessels in the device that you initially started off with, have you been able to do any measurements of gas diffusion or small molecule diffusion across those?
No. Of the intact vessels, or of the
both, comparing intact vessels to the ones that you use.
Yeah. Well, so a lot of people have studied transport across small blood vessels, intact vessels, right? And now it's known that the characteristics depends very largely on size of the vessel and a lot of different things, right? Where first of all, the intactness of the endothelium, but the endothelium is going to be intact for the actual vessel, right? On the second part, this is, we kind of keep this as a very generic tubular construct right now, right? So then the first question is, what size? What's the compare? What's the structure, right? So this is work that's yet has to be done, right? There's a lot of work that yet has to be done.
We gotta move on.
Okay, all right. Sorry for taking so much time. It was a pleasure. Okay.